Great. Well, it's really terrific to be here. Really is one of my favorite conferences, and yes, I'm conflicted in everything I'm going to tell you today. Okay, so I'm going to start with the theme that you've heard many, many times already, which is that medicine these days is largely focused on illness, and like many, many others in this room, we want to really be focused on health and keeping people healthy. What is health? Keeping people healthy. Be very proactive and using as much data as possible to be able to keep people in a so-called healthy state. Generally, the way medicine's done now, you're not measured very often when you're healthy. And I would argue the way you should get measured should depend on your disease risk and how your trajectories go, building personalized models for your progression through various health states. And so I think this is the direction we want to go. And of course, like everyone in this room, we want to be very, very individual based in our decisions for making decisions about health. So many things impact your health. Your genome is one, and lots of folks are studying that. And then we tend to study lots of downstream effects of health, say biomarkers and things. I think an area that we're only getting good at, that's a theme coming up a lot in this conference, is having other mechanisms, devices, say wearables and such. That'll be a big part of this for capturing other parts. So lifestyle, now you can capture with wearables, I'm sure you appreciate. And I would argue a big hole has always been trying to figure out what is your environmental exposure at a personal level. And so these are areas that have interested us, and I want to spend some time on that today. So many of you probably know that a number of years ago, actually 9 now, this month, we set up what we call personal omics profiling, where the idea is to try and use data to try and understand what it means to be healthy and then catch people as they transition to disease. So we study their genome once, do that once, but then we actually make, in its most deluxe forms, 14 different omics measurements over time, typically through quarterly visits, and then much more frequently during, say, perturbations such as a viral infection. We have people doing dietary perturbations, colonoscopies, what have you. And these days, it's around 107 people we'd followed roughly for 5.5, 6 years, some more, some a little less, some more newly enrolled. But the idea is to do big data to follow them. And as I say, I'll spend most of my time talking about some of the wearables work we're doing that we probably launched about 5.5 years ago, something like that. And I should point out that this cohort, it's an older cohort, it's about 54 years old. They're really well phenotyped for diabetic measures because I'm actually type 2 diabetic, which was discovered through this study. And so we have them classified for insulin resistance, OGTT, things like that. There's deep clinical testing and questionnaires. So again, we follow these folks over time. And just to give a quick summary of some of the things we've learned, by following this group, say, through the first 4.5 years, we actually found 47 major health discoveries. Now, not counting hypertension, if you add that, you go up to 67. In fact, by the new rules, you even go higher for hypertension. So pretty soon we'll all be unhealthy. But anyway, these are things that, a lot of these are extremely big deals. 12 people learned something clinically actionable from their genome. So as an example, one person had a cardiomyopathy mutation, and actually turned a young guy, turns out has a heart defect that was discovered through this, and actually is on medicine as a consequence. And then there's other cases. I'll show you an example in a minute. One person we caught with early lymphoma showed up, as I say, quite early. 2 people had precancers, MGUS, smoldering myeloma, things that are fairly important for people to know. Another person with heart issues and so on. So the point is, by using big data, I can't say exactly which disease you're going to follow, but if you're following an older group of people, over time, you actually will learn important information that is valuable for managing their health. And so this is a research study, but I could envision some form of this actually working its way, becoming a clinical test. So just to give you 2 examples, this is an early lymphoma. This was discovered by actually imaging within a large spleen, although there were several other biomarkers that went up at the time. And in fact, we even found a new one, that started coming up about 12 months before the imaging was first discovered. Find this, and upon remission, that marker went down. This is one thing that we discovered. We're spending a lot of time trying to understand diabetes because I am type 2 diabetic. This is what people thought they had coming into the study. So 9 people thought they were diabetic. It turns out 2 are diabetic who had no idea. And a ton of people are pre-diabetic. And my understanding was 70% of pre-diabetics become diabetic. That's different from Eric, and it might number he gave earlier, so maybe it depends on how they're classified as pre-diabetic. I'm not quite sure. We'll talk about that later, I guess. But what's powerful was as we followed these people over time, 9 people became clinically diabetic, and others actually had measurements in the diabetic range. And what was interesting was that they were identified as diabetic by different means, say some by fasting glucose, others by hemoglobin A1C, and still others by oral glucose tolerance test, so GTT. So we actually picked it up in different ways, and others actually by continuous glucose monitoring. Get to that in a minute. And when we actually dug in and looked at these people, they're actually all phenotypically different. Some are insulin resistant, some actually have trouble making insulin. I'm actually insulin sensitive myself and make insulin just fine. I actually don't release it very efficiently from my pancreas. So we're all different in different ways. That's a big deal because the kinds of drugs you take then are important for that. So in my case, actually, I'm a metformin non-responder, one of the 20% who's in that. So again, I think learning these mechanisms will be very, very powerful for treating people. One of the most important things we learned is this concept. I think this group probably gets it, but this really hammers it home. We can follow people over long periods of time, and in general, your basic measurements are generally pretty stable. There are some things that bounce around, but most of your measurements are stable. So these are just 12 people of many measurements over several years. That's me, the blue dots. And over 6 years, my dots always cluster together. And if I get sick, that blue dot will still be near me and not near others. Why is that a big deal? That means we all have our personal profiles. If you get up off your seat, we should be able to tell who sat there if we had the right reference by doing swabs, right? But that's a big deal because this is why GWAS needs hundreds of thousands of people, because they're trying to pick up disease versus controls, but they don't have baselines. So it's really important, I will argue, to get your personal baseline so you can find your difference. The difference between you healthy and disease is a big deal to you, of course, and others, but it's smaller than the difference between you and others. And that's why we need healthy baselines and disease measurements relative to that. And I really think medicine needs to go that way. And no other area, I think, personifies this better than the wearables, because there you can capture healthy baselines in incredible ways. And I think Eric can give you the exact number, but last count, there are something like 900 of these on the market. You have a database for these, if I understand this right. So go look that up. They measure all kinds of things, including radiation, which is always 2-fold higher here, although 3-fold higher in a building over there, relative to Palo Alto. I don't understand that, but you can measure all kinds of things, heart rate, activity, sleep, blood oxygen, so on and so forth. So we tested a number of these. I think we got involved about 6 years ago. And we've been measuring these and using them for various purposes. Some of you may know I wear 8 of these devices every day. There's 3 smartwatches, and that ring is not a ring. That's a sensor, of course, and others as well. So and nearly all of these, I think you know, they'll send your data into your smartphone. So your smartphone is your most important health instrument of the future. I think that's something Eric's said for quite some time. So just this part will be a little bit of review for those who've heard it, but we all have different resting heart rates, skin temperature, blood oxygen, and so on and so forth. And so you can capture that. It varies with the time of day, and you can capture all that too. How much you sleep, when you sleep, all this sort of information. And then the goal is actually to find your deviation from baseline. And so for me, one of the biggest deviations is, in fact, my blood oxygen drops on airplanes. This is known. Turns out this is true for everybody. We've only ever found one exception, a flight attendant who did not drop their blood on a flight. So this is San Francisco to San Diego. Plane goes up, and altitude's green, goes up, comes down. There's my blood oxygen. High goes low, comes back up. What's not so well documented is how low does it go? Couldn't find this anywhere. And how long does it stay low? It turns out for me, for a typical flight, I'm 5% or more, 90 or below. And for most of the flights, I'm running between 92 and 94. So why is that a big deal? Well, you also don't find this in the literature, but it correlates with fatigue. That is to say, for me, that threshold's 96. It's a subjective test, but basically you can do a blinded test. Call myself tired is blue, 96 or below, I'm usually tired, 96 or above, usually alert. It's true whether I'm in a plane, true whether I'm on the ground. And you can do a more quantitative test, which is reaction time. It's blood oxygen as a function of response time, seeing a visual cue. And basically, you're faster when your blood oxygen's higher. and you're slower when it drops. So it makes sense. So the reason we think you're tired on airplanes is because you've been working too hard or writing too many grants or partying too late or what have you. It's because they drop the air pressure, your blood oxygen drops, and you get tired. That's why you're all conked out. I might point out that for those you want to know, there's a free app. It's called Barograph. That's my flight just yesterday coming from San Francisco to San Diego. Your iPhone, very accurate, will measure the air pressure in a plane. We're enrolling more people, by the way, so if you want to join our study, feel free to do so. The cool result, and this is now very reproducible, is that this is actually how I discovered my Lyme disease. Some of you may have heard this story. But the backstory is I was in rural Massachusetts helping my brother put up fences. 2 weeks later, flying to Norway through Frankfurt. And on that last short flight, which is a rather low altitude flight, I measure myself all the time. You probably figured this out. So basically on short, low altitude flights, my oxygen usually drops around 96, comes back. This flight had dropped to 90 median value and never came back. And also saw my heart rate was higher. It's easiest to detect as percent outliers, 2 standard deviations or more. I later learned my skin temperature was off. I wasn't noticing that at the time, but I saw my heart rate was up. And this is low resolution. But then later I got a low-grade fever, went to a doctor in Norway, took my blood, monocytes are up, got a bacterial infection, and he said I should take penicillin. Relates actually to your question earlier. I said, no, I think I need doxycycline, which is what you should use for Lyme. As you might imagine, it was tense for a few moments there, and he did give in and actually took it, cleared it up. When I got back, I gave blood, tested, sure enough, antibody positive, even had some antigens around. In a well-controlled experiment, gave blood before I left, and it was negative. So I seroconverted during that time. So the point out of all this is that I was able to find this by these wearables. And now I couldn't enroll my daughter in a study. I'll be perfectly blunt. I know you're filming this. I actually put one of these on her, and every time she's sick and she's on an airplane, her blood oxygen drops. An airplane is like a stress echo, but for your lungs. Okay, it actually is a way to see how your pulmonary function is doing, whether you're sick. So everybody, if you're sick on an airplane, I'm pretty sure your blood oxygen will drop. Okay. So with that in mind, we actually then looked at all the data. And basically, I had 2 years of data time, 73 blood draws where I measured CRP. And every single time my CRP was elevated, once was a Lyme, twice was with viral infection. We now know that's a viral infection. There was another time where I had high CRP. I didn't report being sick, but it's as high as a viral infection. Every single time, you get outlying skin temperature, and this got cut up and heart rate as well. So that is to say you have elevated heart rate and skin temperature when you're sick. Your smartwatch can pick it up. So we wrote an algorithm to be able to tell. We call it change of heart. It doesn't work so well for skin temperature. I think people wear their watches kind of variable in terms of tightness. But basically, every time you're sick, we think your heart rate goes up, and you can pick this up as a delta from your resting heart rate. So it works for me on all 4 times I got sick, worked for 3 other people, one of whom got sick twice, with very low false positives, as far as we can tell. Okay, so now we're rolling this out for 1,000 people. And again, if you want to join that, we just wrote an app and got approval. We want to test that on a larger group. So the other area that we're studying is continuous glucose monitoring. You're going to hear a lot more of this from Aaron Siegel. I'll just tell you what we're up to. We put this on our normal folks and our pre-diabetics and a few diabetics, these continuous glucose monitors, just to see what people's patterns would look like. And much to our surprise, we saw, there were people with fairly normal flat glucose, good glucose control. A lot of people were what we call moderate spikers, and then there were quite a few severe spikers as well. So we wrote an algorithm. This is using spectral cluster to start classifying people by basically their phenotypes. We call them glucotypes. They're glucose phenotypes. And basically, we think there's a lot of information in these curves. And this is just the simplest form of this. But basically, you can capture the information and classify people. Each row is a person. And if you're not spiking much, you're in this low glucotype. If you're spiking moderately, you're a moderate glucotype, and a severe one over here. Most of the time, you're spiking. And I think Aaron will go through this in a little more detail, but we've also put people through standard foods, and like him, have discovered that actually people on different foods will spike differently. So some people will spike to peanut butter and bread, others to protein bars, and so on and so forth. Different carbs spike people differently, as do other things. One thing that was kind of amusing is that 80% of our older cohort spike the cornflakes in milk. So that's like poison for you, actually, if you go out there. So I really do believe that, you think about it, you regulate tobacco, and nobody's regulating cornflakes in milk, but which is probably hurting most people? I'm not sure these days. All right, anyway, the big surprise, at least for us, again, I think this is more commonly known now, was that we put these on people who are normal by fasting glucose, OGTT even, and hemoglobin A1C, and we actually saw people who were severe spikers, spiking just as much as diabetics. And so this is probably a little complicated to show, but this is one way to classify people by PCA. Red means they're a spiker here, green moderate, and blue not so much. And then here's the diabetic look. So here are two people, perfectly normal by all the tests we said, but they're spiking just as much as diabetics. You actually wouldn't tell them apart from the diabetics. And so again, we think there's lots of folks out there who, probably lots of you, who are spiking away, especially right now after lunch, that might have something to worry about. The last thing I want to spend a few moments on is an area that's bothered us a lot, which is how do you capture environmental exposures? Because we know environment's a very, very big deal for human health, yet we don't do this well at a personal level. We do it at a city level, and we know there's a lot of differences. I'll come to that in a minute. So this is obviously also a big deal because your biological exposures affect asthma and allergies, your chemical exposures can be carcinogens, things like that. And so we'd like to know what people are getting exposed to, again, at a personal level. And so we've actually gone at this to look at this for airborne exposures, and I left my exposometer here. I'll show it to you. Also, it means it's away from me, not collecting optimal data. So and I like to keep it charged, but all right, this is this. So this thing is sucking up air at about 1/15 of what you breathe. Under the pump, it's got a filter. It's measuring PM 2.5, temperature, humidity. But it also has a filter, a submicron, that's capturing particles, very fine particles. And then under that is a chemical absorbent that's capturing all the chemicals. So we elute off all the things we can get off the filter and sequence all the RNA and DNA to see the biological exposures. And for the chemical exposures, we elute those off and do mass spectrometry and find all the chemicals you're exposed to. Does that make sense? Good. All right. So basically, we do go very, very deep. At some ridiculous number like 45 million reads off each sample. We had 70 billion reads total. And so what we did, we like to go deep on a few people rather than thin on lots of people. So 3 people we followed. That's me. I've been wearing this thing for now about 6 years. These are the first 2 years of data. This is 1 year of data, and that's 3 plus some months. Lots of very defined experiments you'll hear about. Okay, now it just so happens most of these people are in the Bay Area, but I do travel a lot. I'm the green guy, and I travel all over the planet, so we sample all over the place. And the question is, how big is your exposome? How dynamic is it? What's it due to? Is it due to location? Is it due to seasons? What can you quantify? All right, so first of all, how big is it? In total, we came up with 2,500 species amongst everybody, and for me, it's 2,000 species. For one sample, this sample, when I get back and I analyze it, it'll have 300 species on it, possibly more. But that's a typical number. So 300 species is what you're exposed to, thousands of chemicals, it turns out. It turns out it's also very, very dynamic. This is me, that's 1 year person, that's a 3-month person. So we can group these, this is from the DNA, but you can group them into bacterial exposure days, fungal exposures. Plants, of course, are green, animals blue. And so this is me over time. And basically, you can see there are big spiky plant days, fungal days mostly, some bacterial days, and this is clustering. These are the plant days, bacterial, fungal. Every time I'm in Monterey, I get a fungal exposure. So you can actually measure this stuff. Okay, I don't know what my San Diego exposure is. I should have looked that up. All right, so then you can start looking at this in lots of different ways. So this is just looking at me, but it's true for the whole cohort. You can see by comparing samples, my Asian samples are over here, so there is a location effect. There's also a seasonal effect, so you can see can separate out the summer samples from the spring a little bit and others, so you can start seeing these patterns. And when we quantify, most we can't quantify because I think a lot of it probably is personal exposure. But location's the #1 driver, and season's the #2 driver for your exposome, for biologicals. We drilled into this in a little more detail. We took 4 people roughly living in the same area. I'm the guy in red. I travel all over, so my time points are a bit scattered. They're all sampled on the exact same month with 4 or 5 samples. This is a person who lives in San Francisco. This person lives not far from me in Sunnyvale. This person lives and works across the bay. So what you'll notice is we all have different exposomes. Even if we live pretty close to one another, we get different biological exposures. You can also see which organisms are driving it. What's driving the San Francisco one, there's several things, but one happens to be this particular bacteria, which is found in sludge. And if you've ever been to Market Street, this makes a lot of sense. Okay? You can pick up other things as well. So the point out of all this is that you can actually see what's in different environments and what kinds of things people are exposed to. All right, seasons, another big thing. There's hundreds of things that show seasonal changes. Species that show seasonal changes, these are those that peak in spring, summer, fall, winter. Some of these we knew already, but many were new. See, I'm running out of time. I'll just say a few words. Chemical exposures, thousands of chemicals. There are plastics everywhere. There are certain carcinogens everywhere. Their amounts vary quite a bit. DEET is everywhere, the stuff you use to keep insects off, and so on and so forth. But this is the coolest result we have. Pyridine, which is a nasty chemical found in paint, actually turns out anti-correlates with fungal exposures. So my house is painted by a green guy. I have no pyridine, and I get a fungal exposure every time I get home. So, which is probably fine. Now, if you're allergic to molds, maybe you actually want to paint your house with pyridine. You might die of cancer, but at least you won't be allergic to mold. So the point out of all this is that you can actually control your own environmental exposure. First know what's there, and then decide what to do about it. All right, and here's the bottom line. We all have our own personal exposure clouds. Mine actually has cats, dogs, and guinea pigs in it. No surprise, they're at my house, and so on and so forth. So you can even capture who you're exposed to if you had all their genome sequence, which would be an interesting thing to do as well. I'm out of time. Bottom line is, we think all this information, we've been building personal dashboard, it should be ready to go in a month. But you can actually pull all this information, follow yourself dynamically over time for all these different exposures, wearables, et cetera. And I just have an amazing lab that has been able to carry out all this work. When You Shall, Brian Pining, Kevin Contrapaw, major contributors. The wearable was done by Xiao Li, Jesse Dunn, Sophia, and Heather, I guess, and spells them by Chow and this gang down here. So thanks again for having me here.